everyone. My name is Ilya Belevich, and I'm coming from University of Helsinki. And today in these 10 minutes, I'll give you a quick introduction to Microscopy image browser. If you haven't used it before, so maybe it will give you kind of good idea about it, and you will try it. If you used, maybe you stick to the end, and then you'll see something new. But before I start, I would like to a little bit go into the history of the things. So in 2011, we started our journey into volume electron microscopy, and it turned out to be quite immediately clear that we can't proceed any further if we won't have any efficient tools for the image processing, segmentation, quantification. And that was our that was led us to development of MIB. And then the goal is really address these three steps that we have in the imaging pipeline. MIB itself is the open source MATLAB code, but it also has compiled version for Windows, Mac OS, and Linux. So if you don't have MATLAB, you can still use it. And during these 10 years or so, we developed quite many different tools and things within the software. And here on the left-hand side, you can see a snapshot from the website with a lot of different tools. But I would like to highlight our kind of major thing that we have is variety of segmentation tools. And already from this list, you may spot that there are a variety of manual, semi-automatic, and automatic tools, including training of convolutional neural networks for training, to train them and then predict images. And as you can see here, lots of these have red dots, or if you in magnified view, they look like these YouTube icons, which means that many of those have tutorials, like video tutorials or video examples, where you can see how each of these tools is can be used. It's not just about image segmentation. We also have a variety of tools that could be used for different things. For example, we have different pipelines. This is like example of anonymization pipeline for unbiased analysis of images. You can do visualization of volumes and surfaces. We can do contrast normalization or photo bleaching correction. You can stitching alignment, and of course, quantification measurements. Like few things that I want to highlight today is one of them is this tool for deep learning, because these days image segmentation without deep learning, it's basically a waste of time. But the problem is that it's not really straightforward in many cases. So what we'd like to have at the time, we'd like to have a solution that would allow us to fulfill these criteria. So it should be easy to install. It should work straight out of the box, so no dependencies, no hassle with installation. Then in order to be suitable for biologists, it should be you're not expected to know how to code. And it should be, has quite nice user-friendly interface so that you can still have possibility to tweak whatever parameters you feel important for training. And naturally, we'd like to have it, to use it for both training and prediction of data sets. The tool itself can be started within the Microscopy image browser from this tools menu. And then within this, you'll get the access to different workflows. And then within each workflow, there are selected architectures. And here you can see the list of workflows. So 2D and 3D semantic segmentation, where we can detect the individual classes, organelles. Then there is a patch-wise, when it's more like classification task, when the network predicts which class the patch, which is part of the image, kind of small piece belongs, and then you can do this kind of patched image. And also there is a 2.5D semantic segmentation, where we try to bring the context information from the previous and the following slices, and then arrange them, in this case, as the color channels. And then it gives the network possibility to know more about our 3D data sets. Itself, within the tool, you basically follow this kind of pipeline, starting from one tab to the other one, from the left to right. We can specify in the directories and preprocessing tab, we can specify locations of our data, their format, maybe split files for training and validation. Then in the training tab, we just specify the most critical parameters for the training, such as input page size, selecting architecture and the workflow. And then when the network is trained, we basically go in the third tab, and then we predict the data which network needs to identify. There are lots of different things there. I just want to highlight a few things. So we have extensive augmentation design dialog, where we can specify probability of each augmentations. And then also you can specify a variety of each of this augmentation in order to be in order to extend the training base. The other tool which I show here is the Activation Explorer, so which you can actually look inside of the network. You can see the weights, sort of the filters the network identifies, and see activation, which whatever network produce at each of the layer of the network. So in this case, you can see that the filter is network learn the filter that defines like this diagonal pattern. Like what we can do with this, here's an example of like one of our recent publications. So this is focused ion beam SEM dataset of a human macrophage cell. As you can see, there are lots of structures. Now nuclei comes, and then we can apply deep learning in order to segment various organelles from that. First, you will see the appearance of the nuclear envelope, and then we train 3D network, 3D net to identify nuclear pores, which we can use for clustering analysis of their localization. Then in yellow, you can see ER with the different morphology, sheets and tubules. Then in blue, it's the Golgi cisterna in the center of the cell. Green going to be mitochondria, and these red spheres are lipid droplets. So like naturally drawing this by hand won't be possible, but with deep learning, we can really create very detailed models that we can potentially quantify. So as I mentioned, we do a lot on this with volume EM, and then our tools are really tailored for this. But in general, we also deal with a variety of data. And during this community days, there was a lot of on tracking of objects. And here's my example on the same subject. So these are the cells that are just moving around, and our goal is to basically trace them. And what we can do, we can quickly trace a network. And then in this case, it's pretty straightforward. You basically identify, you train a network to put a spot inside each of the nuclei in the cells. And then quite easily, you can train a network and then use this for tracking. In this case, what we use DeepMIB to identify the cells. Then it generated these masks, and then we were using TrackMate and Fiji in order to trace the movements on each of the cells. And then you can extract all this data and do a variety of plots. And here, for example, there's 3 replicates, and then one, which was the manual segmentation, that's comparing to automatic one. But in general, the problem with all this, there are many tools that allow you to do the generation of trained networks, but then, how we create this ground truth data for training. And these days, the best way how you can do it is applying segment anything model that comes from Meta. And here is just an example from Electron microscopy, where I use this interactive tool in MIB in order to segment a variety of organelles as cell profiles, mitochondrial profiles, and lipid droplets from the variety of different modalities: serial block phase SEM, electron tomography, and focus ion beam. So it works quite well, but the problem with this is, not a problem really, but these tools are work in 2D. But in August, I think last year, Meta released Segment Anything 2, which comes with a very increased performance, and now we can apply it for segmentation of objects in 3D. So here I just again made this real-time screen capture of segmentation of various organelles. So in the upper left corner, we're segmenting mitochondria. Here we're segmenting the Golgi cisterna, here nuclei, and depending how lucky you are, you can also segment even the complete cells with this. So all that is available in the current beta, which is you can find it on the website, but hopefully we'll also be able to finalize it into release within maybe in the beginning of May. Finally, I'd like to thank Electron Microscopy unit. I would like to thank a variety of communities that are very helpful in answering many aspects and questions. And then, of course, our funding agencies and also collaborators whose projects I was presenting during this presentation. Thank you.